Hi, my name is Lindsay Edwards. I'm the CTO at Therapeutics. First of all, I just want to say how delighted I am that Relation is sponsoring AI Stats this year. It's a really fantastic conference and it's a privilege to be able to support it. And also delighted to be speaking here today on the subject of machine learning and drug discovery, why chemistry is not the most pressing problem. Before I start, this is going to be something of an unusual talk for an ML conference because it won't be about ML that much. But I really wanted to try, from a drug discovery background, to talk to you about the really important problems and to hopefully help the whole community focus on the stuff that really needs solving. So let's start with Eroom's law. If you've not come across this before, it's called Eroom's law because Eroom's is Moore's backwards. Just like Moore's law predicts the exponential increase in compute power across calendar time, Eroom's law retrospectively shows that the productivity of the pharma and biotech industries has been collapsing exponentially. Even to the point whereby now the cost of a new molecular entity, or NME, is around about $3 billion. You can see from 2010 onwards things start to stabilize a little bit. There have been various different explanations put forward as to why that is, but generally this looks like a pretty bad thing. Now you might look at this and say to yourself, what are those guys doing over in pharma? This is terrible. I'm going to help them fix it. But it's worth saying that an awful lot of very smart people have been trying to fix this for a very long time, and the problem is much harder than it appears. So what do you get for your $3 billion? This handy figure shows the funnel from, on the left, the identification of your drug target. So this is usually a protein in the human body that you're trying to get a small molecule or some other molecule to bind to modulate the biology. Through the discovery of the medicine to modulate that biology, so that includes compound screening, lead optimization, through to human testing, so phases 1, 2, and 3. Phase 1 is where we give the medicine to healthy people to make sure that it's safe, broadly speaking. Phase 2 is where it first goes to patients to see if there's any signal, is it actually going to treat the disease? And phase 3 is the big study that typically a pharma company will run to really show beyond reasonable doubt that it's safe and effective. But this isn't the whole story. A much bigger part of the costs is just down to failure. The probability of approval from FTIH, first time in human, is only about 6 or 7%. So that means that you can do all of this work, you find a medicine, you find a target, you give it to a human being to check that it's safe, and somewhere between that point and the point where you're trying to get your medicine approved, 95% of your efforts will fail. And that's where the real cost lies. So not surprisingly, there's been a lot of work done looking at why we fail. And this handy figure on the right looks at clinical failures between 2013 and 2015. And to summarize, the two big bits of that pie are efficacy, 52%, and then safety at 24%. So fully, more than 3/4 of failures are explained by efficacy and safety. Now, a few years ago, I might have had to have explained what efficacy means. Because of the pandemic, most people are familiar with that term, but just in case you haven't come across it before, efficacy is basically, does it treat the disease? So 52% of our failures is because we do all this work, we give the medicine to humans, and it doesn't treat the disease. And 24% are we give it to humans, and there's some toxicity associated with it, which is unmanageable. So the first thing is lack of efficacy, and then you have this sort of toxicity or safety issue. Now toxicity comes in more than one form. So we have off-target tox. So this is where your molecule, which you've designed to bind very carefully to this one protein, actually binds to lots of proteins, sometimes referred to as being promiscuous. And so that causes other problems. Sometimes you'll hear pharma people talk about a molecule being dirty, which means it binds to lots of stuff. Or it's on-target tox. So that means your molecule's binding exactly the target that you wanted it to, but it's not, there are additional effects related to binding to that protein that you didn't predict ahead of time. So what's interesting is these two, lack of efficacy and on-target toxicity, are entirely down to the biology. You could have perfect chemistry, and you never have perfect chemistry. You can have very good chemistry, by which I mean your molecule really binds great to your target. It's not dirty. It doesn't bind to other things. And let's say we split the on-target and off-target tox 50-50, you'd still be looking at 65% approximately of your failures, entirely down to a lack of your understanding of biology. So if you add up all of the costs in the funnel, including the chemistry costs, and burn that money 2/3 of the time, that's entirely because of a lack of understanding of target biology, nothing to do with the chemistry at all. And Derek Lowe, who has a fantastic blog called In the Pipeline, which is widely read by pharma and biotech folk, pointed this out a while ago. He said lowering the cost of preclinical stages by 20%, or making them 20% faster, does indeed save you some money, but those are overwhelmed by the savings that you could realise if you could just reduce the clinical failure rate by 20%. The absolute best way to do that would be through picking better targets. Important note here is the clinical failures are not necessarily, and in fact relatively rarely, be careful what I say here, because it does fail because of poorly conducted clinical studies. But a lot of it is down to the fact that we just didn't understand the biology going in. So going back to this slide, another really important point to note here is that all molecules have some toxicity. So it's not sufficient to say that we're trying to get rid of tox. Even water is poisonous if you drink enough of it. What we're talking about here is the range of dose at which your molecule is efficacious, so it works, and at which the side effects are tolerable or unnoticeable. And in drug discovery, we call that the therapeutic index, this sort of window between the thing works and the thing is making me iller such that the juice is not worth the squeeze effectively. And again, that's really important to remember. So before I go into explaining the biology anymore, I thought a quick genetics primer would be useful for those of you who've, it's been a while since you've done high school biology or high school genetics. So very briefly, human DNA comprises of 3 billion base pairs. Now it's a little bit more complicated than what I'm going to say, but you can just assume that as being 3 billion letters drawn from the set A, C, T, and G, split up into 23 pairs of chromosomes. So within this 3 billion base pairs is the code for 20,000 proteins. I mentioned that number earlier on. Used to think it was a lot more. It turns out that that there are genes for around about 20,000 proteins, and proteins are the machines that drive most of life. So the bits of DNA that encode these proteins are called exons, and the rest of the DNA is called, you guessed it, introns, and the vast majority of the 3 billion base pairs of DNA are intronic DNA, so they're not necessarily related directly to a gene. Now, when we first found this out, someone rather foolishly decided to label all of that intronic DNA as junk DNA. It was thought to be this kind of endless desert of useless letters that weren't really contributing anything to life at all. But evolution is smarter than that, and we should have been. And now we know that actually intronic DNA is incredibly important, and it plays all of these sort of complex regulatory roles. So, for example, stuff that happens in intronic DNA regulates how much of the exonic DNA is turned into protein. It also affects the structure of the DNA and loads of other stuff besides. Now, the last technical term I'm going to use occasionally is this phrase GWAS. It stands for Genome-Wide Association Study. And simply put, this is just where we're trying to figure out the genetics driving a particular trait or disease. And to do that, what we do is we take human beings that are similar in almost every other way. We split them into two groups, one group that has the trait or disease and one that doesn't have the trait or disease. And then we just look to see how their DNA systematically differs. And this gives us an insight into which bits of DNA are associated with the disease or trait. The reason I'm telling you all this is because just about the only piece of data we have at the moment that tells us how to get better targets, and remember a couple of slides ago I was telling you that target biology is the big problem here in terms of costs of medicines, is a paper published by former colleagues of mine at GSK, Matt Nelson and others, which showed that if you're able to show from something like a GWAS study that a gene or protein is associated with the trait or disease, and then that gene or protein is a drug target as well, then your probability of success doubles. On the right-hand side is a paper by Emily King and others, which was published in PLOS Genetics a couple of years ago, basically confirming the findings of Matt's group, but with new data. So this is a really useful piece of information. And lots of big and small companies, including our own relation, is interested in the use of genetics to help us find better drug targets. Importantly, though, what that paper doesn't say is that GWAS hits make good drug targets. And there's a bunch of reasons for that. So the first thing is that the vast majority of the GWAS hits, so the things that are systematically different between our two groups, disease and control, for example, are in introns. So it's not immediately clear what that variant is doing in terms of the biology. And figuring that out is non-trivial. The second thing is that those papers that show that genetic support for a target increases its hit rate literally don't say that they make good drug targets. And one possible explanation for that reversal is a similar thing you see. The analogy I've heard drawn before is most Ferrari drivers are men. That doesn't mean that most men drive Ferraris. In this case, most drug targets with GWAS support are successful. That doesn't mean that all GWAS hits are good drug targets. And there are many more GWAS hits than there are successful drugs. So it might look a little bit like this figure I've drawn here. Another really important point is that lots of incredibly successful drug targets like TNF-alpha, I'll come back to TNF in a minute, often don't show up in GWAS screens. And one simple explanation for that is this idea of conservation in evolutionary biology, which is just that if you have a bit of DNA that's really, really, really important, any changes in that region of DNA kill you. And so it doesn't get transmitted. And so if that's the kind of case, you have these highly conserved regions of DNA, and that means that they won't show up on a GWAS screen for that simple reason. That having been said, not only is genetic support incredibly important, but we can use genetics to shine a light on disease-causing biology. Let's quickly go back and focus on those first 2 points, that most GWAS hits happen in intronic DNA, and unraveling the effect that those changes in the intronic DNA have on the biology is hard. Encouragingly, this has been an area where machine learning has made some real progress. And we now have something like a 6-year history to look back on. One way of thinking about this problem is that we're trying to featureize the DNA. So if you think about a region of intronic or exonic DNA, we want to understand how that variation, so changes in those letters, C, A, T, and G, is affecting what happens in that region of the DNA. So that might include the binding of things called transcription factors that regulate the biology. It might include these epigenetic marks, so methyl groups being attached to the DNA. It might change the way that the DNA folds. All of these things are incredibly important in determining what happens to the biology, even though this bit of DNA isn't directly related to producing a protein. And the early models, the first one I've got here is Alifani et al. That model was called DeepBind, and it used a one-dimensional convolutional neural net, sliding along regions of the DNA and predicting whether or not a transcription factor bound in that region of DNA. Later on, we had DeeperBind that I think used LSTMs. It was only a matter of time before people started using transformers as they became state-of-the-art for language modeling. And now we've had a few papers in the last year, one using a GNN, and this paper, Avsek et al., which used transformers to predict transcription factor binding, methylation, chromatin, so this is the molecule of DNA conformation, so the 3D structure, and then layered on top of that are predictions of what might happen to gene transcription, so how much of each of those proteins there is in a cell, roughly. And a lot of that has come out of Calico, but also Olga Trojanska's lab has been doing a lot of work in this area as well. Let's move on to this penultimate point, that many successful drug targets don't appear in GWAS screens. So in addition to the issues I raised earlier on about conservation, another model has also been proposed called the omnigenic model. And the omnigenic model suggests that the genetics of disease are not necessarily gotchas. Sometimes they are, sometimes you'll see there's been some disruption, like a mutation in the gene. It breaks the gene, it breaks the protein, and that clearly causes the disease. But actually what we see more often, and particularly in big, important chronic diseases like diabetes and heart failure and asthma, is in fact that lots and lots of small mutations in genes kind of stacks up and causes problems. So how do we deal with that? There was a very influential paper, I think, last year from Julian Knight's lab, not really a machine learning paper, but in this paper, they were trying to aggregate information across sort of regions of biology. So in this figure on the left, remember I mentioned TNF before, this figure shows that lots of the genes that interact with TNF, you do see mutations in them. And there's this sort of guilt by association process that you can infer. Now, in this paper, they used traditional network methods, but to us at Relation, it seemed like a no-brainer to use GNNs. This is the bit where I don't really need to tell this crowd very much about GNNs, but they are powerful tools for aggregating information between genes in this case. So the message passing model allows the architecture to share information between genes, and it seemed like a natural choice to aggregate the different streams of information that were being aggregated in the Julian Knight paper. And we found that when we used GNNs for this task, rather than, for example, boosting that was used in the original paper, we saw a pretty significant performance boost, around a 70% uplift on the average precision score across multiple indications, compared with what they saw in the Julian Knight paper. This hasn't been published yet, but we're working on the paper now. So another area I want to call into focus is around manifold learning for biology. And this is based on ideas from the late 90s. So my background as an academic was in systems biology, and one of the seminal papers was this one here, written by Trey Eidecker, Tim Galitsky, and Leroy Hood in the early noughties. And the basic principle was this: if you take a cellular system or a living system and you perturb it, so you sort of push it in lots of different directions, and then record what you see using something like imaging or some method for measuring all of those proteins, what you hope is that you'll eventually see everything that cell can do, and then you can fit a manifold to that, and that will allow you to make predictions by doing vector arithmetic on the manifold in a way very similar to what you see with something like Word2Vec, for those of you that are familiar with that work from some years ago now. Sounds great. The problem, however, seems to be that biological systems are dastardly in their ability to not show you lots of their behavior. And if I had to pick one thing that trips most people up moving from physical sciences or things like mathematics into biology, it's massively underestimating just how complicated even the most simple systems are. So in this case, what tends to happen is you have a cellular system, a bunch of cells, you've done loads of stuff to them, you think you've seen everything, and then you just poke it in the right way, and all of a sudden this cell has all this other behavior that you've not seen. Another big problem that we've seen is to do with data quality. So I certainly spent several years chasing my tail, trying to remove systematic biases from data sets I'd found in the public domain. And again, it turns out that really is pretty much a waste of time, that you use a lot of your model's expressive power dealing with data issues, and you almost certainly need to go and get some new data. And there are a bunch of companies now, including ours, who are founded on this idea that getting data is incredibly important. And in fact, at Relation, we've gone one step further. We have this idea called Lab in the Loop, which is where not only are we gathering data, but we're using active learning to allow the models to tell us what data that they need. And in order to do that, we are building our wet lab and our dry lab and our machine learning and our biology teams completely tightly coupled. So everybody sits together and works together on these problems. We're not the only ones talking about the importance of active learning for discovering new biology. There's a long history of doing this, actually. But more recently, Caroline Euler had a very nice paper, this ABCD strategy, using Bayesian design of experiments to pick optimal batches of experiments to run. That's a very important consideration here. We don't want to run experiments one at a time. And at ICLR this year, Kim Branson at GSK and his team, in collaboration with Yaron Gal from the Oatmeal Group at Oxford, are sponsoring a challenge at the Machine Learning for Drug Discovery workshop, again encouraging people to find new active learning methods to discover new biology. And I would say this is a really, really good time to get into this space if you're interested in doing something worthwhile in drug discovery. Thinking about active learning for sort of undiscovered biology is a really good space to get into. If you're interested in what Relation is doing in this space, we recently open sourced our Pi Relational package. So equally, if you wanted some nice tools for doing this, please do download the package and use it. Please do contribute. It's all welcome. Briefly, I wanted to touch on one other thing, which is a lot of the focus in the machine learning community towards drug discovery has been on chemistry. And I said at the top of this talk that I was going to explain why that maybe isn't the most fruitful thing. So the first thing I would say is this paper by Connor Coley. I think Connor is speaking at the Machine Learning for Drug Discovery workshop at iClear as well. But I would heartily encourage you to read this paper. It helps explain some of the problems. But put briefly, chemical space or chemical spaces are incredibly complicated and dense and massive. And the data that we have, even in these large databases like Zinc and Kemble, are the result of human beings investigating stuff. And so what tends to happen is you'll get like a puddle of data where everyone's got very excited and looked at a particular type of molecule, and then another puddle of data where everyone's got very excited and looked at particular classes of molecules, and nothing in between. And so if you train an autoencoder on that data, for example, the low entropy regions of your model will be in these regions where there's lots and lots of data. The spaces in between are pretty much unknown. And if you're using a generative model and you sample from that generative model, what tends to happen is the model will produce a molecule that's incredibly similar to ones it's already seen in training. Going back to our old friend Derek Lowe, he had this to say: if you took any of the AI-generated molecules from these projects and showed them to an experienced medicinal chemist, the response would be, well, yeah, sure, and probably also, you better check to make sure those are actually patentable. So that's in addition to the fact that even if you do solve these problems and reduce, for example, lead optimization, you're going to be having relatively minimal impact on the overall problem of attrition in drug discovery, which is much more related to the biology. So if I had one take-home message from this talk today, it would be: get stuck into the biology. Those are the problems that need solving. They are harder. The data is in a mess, but that doesn't mean we shouldn't be doing it. And I would heartily encourage you to get in touch if you're interested. Incidentally, we are hiring at Relation if you want to come and work on some of these problems. But yeah, I would encourage you to get involved in the biology, and I'm hoping over the next few years to see the machine learning for biology efforts massively dwarf the machine learning for chemistry efforts so that we can make some real progress. Briefly, I just wanted to speak to the role of supercomputing in all of this. One of the huge breakthroughs recently in machine learning for biology was clearly AlphaFold, and the compute resources available to DeepMind are massive. Otherwise, they wouldn't have been able to train that model. And it's not just the training the single model. Of course, it's the research efforts of trying lots and lots of large models and being able to do that in a reasonable amount of time. However, supercompute is available to us mere mortals. NVIDIA recently built Cambridge 1 in the UK. It's the UK's largest supercomputer. I think it's one of the largest in Europe as well. We're very fortunate at Relation, along with 3 other UK startups, we actually have access to Cambridge 1. And so we're also working on some of those language models for featurizing DNA. And we're really hopeful that access to this kind of supercompute will allow us to, for example, increase the receptive field of those models. So be able to look at much more DNA in a single hit, or increase the precision of the predictions. These two things sort of interact with each other to increase the memory footprint of the model. We're really hopeful that Cambridge 1 will help us with that. Watch this space. I will report back if it works. Lastly, this is as a sort of useful takeaway slide, is the current consensus for modern drug discovery. And so we've talked a little bit about genetics. We've talked a little bit about variant to gene mapping. I'll come back and briefly explain what that is, just so everyone's clear. It's important to say that you always have to start with your patients. So most diseases are actually have been categorized many years ago on the basis of the symptoms. When Patrick Vallance was at GSK, he used to like saying that many of these diseases were categorized by Victorian gentlemen in top hats. So things like asthma was a set of symptoms, but we now know that underneath those symptoms, the molecular biology, so what's actually happening in the disease, is different between patients. And so it's really important to understand that. So having access to patient data is really important, and then being able to really understand which patients are like which other patients before you run your GWAS studies. Then when you run your GWAS study, you identify some of that genetics. As we talked about before, the problem with that is that lots of those gene hits end up being in intronic DNA, so you have to figure out what's going on. So that's sometimes referred to as variant to gene mapping. So how are these variations in intronic DNA affecting the actual genes and gene expression, and how much of the proteins there is? Once you do that, you start to get a better understanding of your disease. You then have to spend some time kind of unpicking that. I've called this causal biology discovery, but if you remember the kind of map with TNF in the middle, sort of trying to figure out how these different bits of biology interact and whether there's a big node in there that you might be able to poke with a new drug. You then have to do your discovery of new molecular entities, so figuring out what the molecule is that you're actually going to give to humans. You then need to test for safety before you finally go to the clinic. And this is, I would say, a consensus at the moment. So there's lots of scope here for machine learning to have an impact. But hopefully I've persuaded you today that the really, really critical thing for us to understand right now is biology, and especially target biology. So that's it from me. Huge thank you for your attention. Again, absolutely delighted to be here. Delighted that Relation are sponsoring AI Stats. We are hiring, so please do get in touch. If you found any of this interesting and you'd like to come and see if you can help us solve some of these problems, we would absolutely love to hear from you. Come to the booth, drop us an email, and I look forward to hearing from you. And also, if you have any questions, I love questions. So again, please email me. My email address is Lindsay. That's Lindsay @relationrx.com. That's Lindsay @relationrx.com. Thanks for listening, and enjoy the conference.